Hey guys, what is going on? It's Alexander Williamson here with The Secret History Living in Your Aquarium, featuring Fishtory and also on the Aquatic Morning Show. This week we have episodes numbered 156, 157, 158, and 159. So strap yourselves in, grab a snack, and, well, if you're watching this on the morning show, grab some coffee or whatever wakes you up. But, hey, I don't judge. Anyways, so number 156 this episode. Why fish look downwards when they swim. Apparently, most fish, when moving forward, their eyes focus down on the ground. And researchers wanted to know why. They wanted to know why so that they could implement it maybe in robotics, maybe so that they could figure out maybe there's a neurological reason, maybe there's a reason that it could help vertigo in humans. Basically, motion sickness and other things, they thought maybe it plays a role in that. So here's the mystery. There is a fish that is always brought up on this channel, and it's always brought up in scientific studies, and that is the zebrafish, or the zebra danio. And this fish is used as the prototypical creature in research. And as far as vertebrates go, it's up there with the most studied, along mice and rice fish. And then you got to go to invertebrates and insects, things like fruit flies after that, to find something that's more studied. So, before we jump into the full episode, I do want to say thank you so much to members. Your subscription means a ton to me. And if you're just watching this also on the Aquatic Morning Show, or if you're watching as a member who gets a sneak preview of all the episodes in this audio format where you don't have to watch the screen at all and see how cold it is where I am right now. Anyways, thank you so much. And it's $1.99 to sign up and support the channel this way if you'd like to. Anyhow, back to the mystery of the danio fish. So now simulations show that fish look downward. Not just the danio, but a ton of fish. In fact, most fish look downwards at the riverbed that they're swimming above when they're moving forward. And if there's any current, it also happens. So unless it's perfectly still water, their eyes are mostly looking down. Now, they can quickly dart up if they see danger or a shadow or something like that, and they actually have very strong peripheral vision. Unlike us, they have peripheral vision that is a lot more sensitive in shape and speed and what's going on. So they can dart away from looking at the ground very quickly, which is also important. But the study wanted to know why in the heck are fish staring at the ground like this when they move forward, when they have motion tracking software look at their eyes and they film it. They're seeing that they're looking downward. And it turns out that it's an adaptive behavior that evolved to help self-stabilize the fish when swimming against the current. So they kind of word it as such, which is that as water moves, fish are constantly trying to stabilize themselves in order to stay in place. So rather than getting swept away down the stream or not realizing where they're at and slowly losing speed where they're trying to hold steady, they focus on the ground. They focus on other fish, plants, debris, rocks, shells, sand, whatever's there. Because if they were to focus on moving things like leaves or the water above them, the clouds, the light, whatever it may be, those things all move and play tricks on them optically in the water. So it's similar to like when you're sitting in a train car and you're not moving, or you're parked in a parking spot and your car's not moving, but the one next to you starts to pull out, and all of a sudden you feel like you're rolling forward because the other one's going back. So just like that optical illusion triggers our human minds, which are obviously a little more complex than the danios, they have that same problem. And so this helps them override that when their eyes are trained on essentially the equivalent of how we train our eyes up and on the horizon, or when we're driving, you're taught to focus your eyes farther ahead, like 100 yards than where you're actually driving. This allows them to get the most reliable signals from the bottom of the river. So the study is published as of November 2nd in the Journal of Current Biology and Organism Development. And they figured all this out by using high-speed cameras and equipment that could track the pupils of the fish. And they visited a bunch of sites in India as well as in Thailand to study shallow river fish, and then compare in the lab fish that were zebrafish. So the field team encased a 360-degree camera inside a waterproof diving case, and they attached it to a remotely controlled robotic arm or a little submarine, and they used it to go into the water and swim with the fish and watch where their eyes were going. So it allowed them to put their eyes, so to speak, their camera, where the fish would be, and so they're seeing what the fish would see, and then they took that data and they modeled it in the robot, and they actually gave that to an operator trying to keep it steady moving in the water when they're trying to film, because apparently this is also an issue when they're filming underwater, or when they're trying to keep something steady, like working on a repair on a pipe or something underwater. You can get tricked as the operator really easily, apparently, and divers are subject to this problem too. They get tricked, and sometimes you get confused which way is up. Even if you've heard of diving, you know that the water sometimes has different salinity and things, and now they're realizing that just the simple task of putting the bottom of the water in the data feed and mapping that and then stabilizing that can give a horizontal baseline for all the work that they need to do underwater with this kind of stuff. So it's actually going to be pretty useful and kind of interesting. I hope it doesn't get used by oil pipeline folks and the work they're doing underwater and then kill a bunch of fish, but that remains to be seen. So that is today's episode of Fishtory. I hope you all have a wonderful day. If you guys are watching on the Aquatic Morning Show, I hope your week starts off well. Well, so do I for everyone else listening to this too. But wanted to say hi to you guys in particular. Hopefully I'm in the chat hanging out with y'all. Probably not, because I'm procrastinating making this last minute recording the actual speeches that I wrote. So I will see you guys later this week, and to everyone, have a great day. Bye. Hey everybody, it's number 157 of Fishtory, and that is how fish populated ancient oceans. It's not what you think. So you may assume that when new species came onto Earth and they took over the oceans, that maybe they started in the shallow waters, like where single-cell organisms and algae and stuff live mostly, and that it progressed, you know, along shorelines and then just kind of made its way on coral reefs and shallow rocks and things like that, where we see most life today. And so evolutionary biologists thought for a long time that they probably just colonized the shores first and then got deeper and deeper. And over time, a few, you know, daring evolutionary steps would take them to things like the sulfuric vents and the deep waters. And they consider, like, deep water in marine biology anything over 650 feet, where there's no longer sunlight, or the abyssal zone, and deeper. So that means that there's a lot less food down there. It's not as warm down there. And so it makes it a difficult place to live. So they figured that, you know, that's not where fish would choose to go first. However, by analyzing new relationships using the genetic records of fish living in these deep places, as well as the fossil record going back over 200 million years, University of Washington, today, Junior, I'm stuttering, sorry, 200 million years ago, we're talking, and a University of Washington biologist put this all together with her team. Her name is Dr. Miller, and she was able to identify a really interesting evolutionary pattern, and that is maybe a lack of pattern, and that it is that new species evolved very quickly in the deeps, very quickly after fish started even going into the deeps. So it wasn't like we thought, like there's this huge diversity in the shore and then nothing in the deep, and then slowly more has built up in the deep over time. Actually, things built up all over the place, and they're trying to figure out, why that is, what happened, and why in some cases there's rich diversity in the fossil record of deep ocean water and not as much in shallow water. And it turns out that there's a number of reasons this happens, and this discovery raised questions on like what those reasons could be, and what causes a fish to prefer one habitat over another, and what made some fish able to move into deep water easily and others not even to be able to survive in the food-rich shallows. And so this will definitely inform us of our species diversity today and maybe what the planet holds in the future. So Miller mapped out these flip-flopping speciation rates onto a timeline of Earth's history, and she was able to identify 3 major events that probably threw all this for a loop, instead of it happening the way you'd think, where all the food and light and everything is. Things evolving rather than down in the dark, deep, cold places. And the first was the breakup of Pangaea. So the supercontinent of Pangaea, which was all of the continents kind of smashed together 200 to 150 million years ago, that created coastlines all over the place that may have been really diverse, but when it split up, it started to isolate fish populations, and also fish had to move from shallow to deeper water. There was suddenly a lot less access points to those shallow areas, instead of going around all that continent and just following the edges in different temperature zones and things. Now fish could journey across the water and try to make it to the shallows again, but some inevitably in that journey in open water and connecting new spaces decided, hey, let's go down, let's try it out. So they think that's the first thing. And the next one was the Cretaceous hot greenhouse period, which all occurred around 100 million years ago and marked one of the warmest eras in Earth's history. So during this time, many continents were flooded due to the highest sea levels Earth has ever seen. A large number of new shallow areas across the Earth and low water fish diversity all of a sudden take off, and you see all these species that we see in the fossil record that then go extinct and their lineages don't continue. And so when the sea receded again after that, fish that were in the depths did fine, but the ones in the shallows had to keep going back and had to keep competing with the change, and probably didn't do as well. So the third major event was a climactic change around 15 million years ago, known as the Middle Miocene climatic transition. And this was caused by further shifting of the continents, which caused major changes in ocean circulation and cooled the planet. It basically allowed all the jet streams and currents that we know today to have formed by having the Atlantic and the Pacific Ocean and these big barriers of North and South America in the way. And around this time, we see deep sea speciation rates speed up the fastest we ever do in 200 to 300 million years of data. And it's driven by the cold water fish that live in the poles being able to follow the shore, but also be able to follow the cold, and so in the tropics they go deeper. So climate's not the only thing that probably explains this, and not every species has the right combination of traits to survive in deep water or start, making that transition. And so it's kind of a limited area without the sunlight. New systems such as sight need to evolve, or touch, or senses. And so fish like catfish, you can think of with their whiskers sensing motion, and the fact that fish have things like lateral lines and traits that we think of as common in shallow fish today actually probably evolved in the deep for them to sense water pressure and things around them, and those were just really handy when they came back towards the shore and split off into new species, which makes sense, and it's kind of interesting. So right now, I don't know that this research is fully fleshed out or anything like that, but it is super interesting because the deep sea creatures also were the first to have like big jaws and sharp teeth, and were able to hunt bigger animals that were out foraging, you know, in the open waters, kind of like today we see tuna and whales and things like that in open water, and less of the diversity and tiny things that live in the shallows. So it's just kind of interesting. Also, when those currents started up 15 million years ago, it allowed water current patterns that also allowed animals to use those currents to scoot along the seafloor and follow that temperature of water, the pocket of water they were in, and actually like keep feeding year round in a semi-similar climate, or temperature rather. I guess it's climate underwater, to the one that they were used to. So all these things are just little things that made me think, and it came out of the university right next to my house in Seattle, and so maybe I'll have to go over there and talk to Miss Miller, Dr. Miller that is, and ask her some more questions about this. Maybe we can get her on the show. Who knows? We'll try. All right, guys, have a good day, and I'll talk to you mañana. Bye. What's going on, fishy people? We are on episode 158 of Fishtory, and today we're going to be talking about the Devil's Hole pupfish. Yeah, you know, a little late for Halloween, but turns out new research says that they are the most inbred fish that we know of. In fact, they're probably the most inbred vertebrate that we know of. So researchers over in jolly old England compiled the first genomic sequence of the desert pupfish, including the Devil's Hole pupfish, and they looked at pupfish from the American Southwest, including those unique Devil's Hole pupfish, and the genomes of 8 Devil's Hole pupfish that they sequenced after they had passed away of natural causes, and they found an amazing number of identical stretches or segments of DNA. In fact, 58% of their genome was identical segments, up to 15 different times, and also, paradoxically, there's over 15 gene deletions from other fish that we know that they evolved from those fish originally, and they had certain genes, but they got rid of them. And one of them is the gene that allows them to live in low oxygen or hypoxic water, and the other is one that allows them to live in hot water, which is not convenient considering they live in Death Valley. So it's really odd that they dropped those genes, and it could be inbreeding that did it, and honestly, it could be inbreeding that signals an end to the Devil's Hole pupfish, unfortunately. So then after the Royal Society of Ichthyology got their hands on that genome, the folks over at the University of California, Berkeley, UC Berkeley, also were sequencing different individuals, and they had old species from the 1980s, and they had 30 new ones, including 15 of Devil's Hole specific pupfish, and they said that of the 8 individuals that were found live, or not found live, but recently died, that were living in today's era, they found 58% of their genome was the same. Now, they also had specimens from the 1980s that were preserved, and they were able to pull their genome, and they saw that about the same number, about 57% of their genome was the same. So inbreeding in the Devil's Hole pupfish is probably not something like drastically affected by humans. They've probably just been cut off for a really long time. You know, it's not often that you see this inbred of genetics. Apparently the African mountain gorillas and the Indian tigers these days are seeing very, very inbred rates, but that's due to human activity and them being kind of hunted into these small pockets for the last 200 years or so specifically. So it doesn't look like there's an increase in the inbreeding due to humans necessarily. If they had like 100-year-old specimen, it would be even more compelling to show them that. But other pupfish species are also inbred, and a lot of research that has been done has found only 10 to 30% of their genomes are the same within a species. So with the new genomic data, there is a lot of potential to look at not just genetic diversity and how these species are related to each other taxonomically and phylogenetically, but also look at inbreeding and the mutations that must be going on to allow them to keep surviving even though they are so inbred. In fact, they're so inbred that it's the equivalent of them living at least 6 generations and only reproducing with their parents or their siblings. And surprisingly, as bad as that sounds, it should be a lot worse because they've always had a population, as far as we know, in the Devil's Hole pupfish population that's about 300 or less, and they've been in one little body of water, so there can't be that many lineages. So there's something going on there that is somewhat stabilizing the genetics that are needed and essential, but at the same time, the Devil's Hole pupfish seems to be lacking that anoxic gene, and it could be why we see big die-offs in their numbers. So sometimes those fish die off to like 40 fish left, and other times there's 300 of them the next spring. So it's really kind of a mystery what happens to their population, and we've been trying to figure it out. But now they're saying that we might be able to take the genetics and the genome from a similar pupfish, or even from the ones that we took in the 80s and put into Site 2, because there's like a man-made site that also now keeps pupfish, because we couldn't figure out how to keep them in captivity, actually. And there's also populations of closely related pupfish on San Salvador Island in the Bahamas. And so in San Salvador, they're trying to study that one too. And they're thinking of actually being able to splice some of the DNA that's missing about like their ability to withstand heat. And it could be that that's recently new developments, because during the Ice Age and things, they probably were doing fine in there, and then Death Valley became what it is today over the last 10,000 years or so, and it may be that the water has stayed somewhat stable, but now it's reaching a point where it's just getting too hot. So they're hoping that maybe they can use technology like CRISPR and cross in that gene for hypoxia that was once in their population at some point anyways, probably before they got into that hole where they all live now. And they're thinking that maybe they could maybe save them that way. And then other researchers are saying, like, no, what are the ethics on that? This fish has evolved, and maybe its time has just come. So I'm curious to think what you guys think about that. Like, if we can save this species, but it only lives in one hole in the desert, should we do it? Do we do it just for preserving them and maybe breeding a kind that we could keep in captivity if we can figure that out? So kind of big ethical questions here, but it does appear that it may be a way to save the species and actually restore those genes that were lost and restore that diversity, giving them different lines and kind of refreshing their population with their own genes even. So some people are saying it's unethical to take another species and cross it in, and that would be a hybrid. But if you could rearrange using CRISPR, their own genes, or basically genetic material made out of star stuff, made out of proteins and things from their own GATC sequence, then that would be more ethical. So I don't know, is it more ethical to save them if we use their own genetics and cut and paste, copy, alt, delete, whatever? So I'd like to hear what you guys think, and I will see you on the last episode of Fishtory this week, tomorrow. Have a wonderful day, and I will see you guys tomorrow. Bye. Alright guys, we are on episode number 159 of Fishtory. I hope you guys are doing well. I hope your week is off to a great finish. I hope it's gotten to a great finish. So, what I wanted to talk about today is a little bit of tooting my own horn, in that I covered this story and I had a showdown that has become a bit infamous on my channel. Didn't anticipate it happening, but it's something that made me very frustrated, and that was a certain person I was interviewing laughed and said, "Ha, a woman couldn't have invented the aquarium. Women weren't even allowed to do anything without men in 1870 or the Victorian era." It was a guy, I can't remember his name. Quote, "Well, that's not true. Jean Villepoul Powers, the French ichthyologist, ecologist, and snail studier," I don't know the name of what they are. She was born in the late 1700s, and in the early 1800s, she became one of the first people in correspondence letters to other naturalists, mainland Europe. She became one of the people to make the first glass aquarium, the first four-sided glass aquarium. And she also came up with the idea of, hey, let's take everything that's out in nature with these creatures. If we're studying an octopus, let's go get the salt water, let's get whatever we see they're eating, let's get everything that's on the reef with them and plop that in the tank, and then sit back 20 feet and take notes. Because that was the next closest thing that they could do, short of diving and observing like that, which they also did by swimming in the Mediterranean. But I did an episode on this. I was told that the episode was not who invented the aquarium, and I had a little bit of an outburst and may have been a little bit rude to somebody that I felt was being rude. It's all old history, and most of you probably know who I'm talking about. He's a young kangaroo, if that helps figure out the name. But what I wanted to talk about is that there are 4 books that have come out on Amazon that I've just noticed, all talking about this lady, and I had to really scratch the bottom bowels. That doesn't sound good. I had to scratch the butt of history. No, I had to really search historically to find good evidence and info on this woman when I made that episode 5 years ago, and now I find it to be pretty common. So I don't know if I had any part in that, probably not. But the story's out, and now there are 2 kids books out about her, and then there is an entire full-length book about her that just came out about 2 weeks ago, and I really want to get a hold of the gal who wrote it. And then there's another book that talks about the start of ecology and how she really started the whole field of ecology, in the sense that before we studied specimens in jars, or we studied certain things, and we studied taxonomy and individual organisms and drawing them in great detail and their anatomy and stuff, but we didn't really look at them in the natural system they lived in. And so there's a book by Evan Griffith called Secrets of the Sea, the story of Jean Powers and the Revolutionary Marine Scientist. There's another one called Matriarchs of the Sea. And then there's another book called The Lady and the Octopus, How Jean Villepoul Powers Invented Aquariums and Revolutionized Marine Biology. And then there's another title I noticed in the UK that instead of marine biology, it says ecology. So I found this interesting, and I want to encourage folks, if you're looking for an interesting read, her story was really fascinating when I was trying to piece it together. And I just got a couple new aquarium history books that I'm reading at the moment, but I'm definitely going to pick these up. Well, not the kids' books. I probably don't need the picture book on this. But I think it's great to encourage young women to get into ichthyology and the sciences. And I think now, this generation, finally, there are quite a few women getting into the sciences. But historically, you know, it's been a boys' club, and we need all perspectives in the science and in the sciences in general. So I think it's great to have role models that look like you, that are, if you're a little girl, it's a woman. If you're a little boy, it's a dude, Indiana Jones. You can pretend to be Indiana Jones as a little 6-year-old kid running around with a whip. Maybe you'll get into archaeology. I don't know. Not that I know anything about getting into archaeology and making it your degree and then never doing anything with it other than kind of like a public access channel newscast thing about fish. Anyways, so back to the story. Yeah, there's a lot of books out about women in STEM, and the glass ceiling that she broke, Jeanne Villepreux Power. She came from a peasant family in France, married well, but then on her own accolades became famous. Unfortunately, her ship sank when she moved from Naples back up to, I believe it was the UK, but I think they were going to France first. Ship crashed. Her life's work, all these beautiful sketches, and they know they're really intricate, beautiful sketches because of the correspondence letters she'd written. So a lot of it was at least duplicated in some degree to other naturalists around Europe, because she spoke like 7 languages and wrote 7 languages and was able to write these letters. And that's now how we kind of have pieced together her work. She also did become the head or president, they had a different name for it in France, of the Biological Zoological Society and Marine Biology Society in Edinburgh for a number of years too. So big accolades at the time. When she started out, she actually had to pretend it was her husband, this Duke guy from France, doing the work. And so she would sign his name, and before she kind of broke the glass ceiling by showing up at one of the meetings of folks in Rome, and then again in Paris, and was like, hey, it's me, I've been doing the work. And they're like, holy cow, you have, awesome. And it was good timing, because the French Revolution was just finished up, and the whole egalitarianism, women and men being equal thing, those crazy revolutionary feminists thinking that women had brains like men were kind of just getting popular in the elite or aristocratic society. Anyways, I'm going to stop yammering, but there's a number of books that I'll link in the article that you guys can check out, or you could just search Jeanne, 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 Villepreux, Villapreaux, I believe, and then Powers, like superpowers. Alright guys, I hope you have a great week, and I'll see you next week on Fishtory.